we talked about germline predisposition as a context, liquid biopsy. Really, part of the session is starting to sort of impinge on these new data modalities and opportunities, including the functional screens. Single cell is probably one of the most avidly and frequent requests we get within CBTN. And I think it's a privilege for me to introduce Aaron Diaz, who's really been one of the earliest users of CBTN samples for data generation, and really leaned into sharing and partnering with CBTN broadly on the single cell data projects because they are so rich and complex.
Thank you, Adam, for that introduction. And thank you for the opportunity to speak to you today and share with you the work that we've been doing using single cell profiling of clinical specimens. Since its inception in 2015, my lab has contributed genomics and functional studies of pediatric and adult brain tumors. And I'd like to share with you today some of our ongoing research, unpublished research into medulloblastoma. And we're performing a longitudinal study of clinical specimens, trying to understand medulloblastoma's evolution under therapy. And I'd like to share with you some sequencing data, but also some genetic lineage tracing data in a model that we've been developing. So here's a genomics overview of the specimens that we've collected and analyzed thus far. We've looked at 57 frozen specimens, 32 FFPE specimens. A critical cohort was provided by CBTN, patient-matched, treatment-naive brain tumor specimens, and recurrent patient-matched tissue as well. And these will be critical for understanding the longitudinal evolution of this disease. We've subjected these specimens to single-cell RNA sequencing that measures a transcriptome-wide expression profile for each individual cell within a specimen. Also, single-cell attack sequencing. This is an assay that allows us to interrogate regions of open chromatin. We can scan those regions for the recognition motifs of known transcription factors, and in so doing, understand what transcription factors are binding where. So the RNA assay tells us about cellular phenotype. The attack assay tells us about transcriptional regulation through upstream transcription factors. We've also been doing some spatial profiling, but I won't go into that today. This is an overview of the first 36 samples that we sequenced, and it's the basis for the subject that I'll share with you today. And I want to focus in particular on group 3, group 4 medulloblastoma. There's been some recent single-cell studies from the Taylor and Northcott labs that have identified the putative cell of origin for group 3 and group 4 medulloblastoma. There's a structure known as the rhombic lip, which is a transient structure that occurs during the development of the cerebellum. And under normal conditions, it's internalized into the cerebellum. Sometimes it fails to internalize and becomes a source of neural stem cell progenitors, which can then go on to lead to group 3 and group 4 medulloblastomas. So those recent studies using single-cell profiling from Taylor and Northcott have described the ectopic expression of oncogenes as a source of genetic insults, which can then lead to failure to differentiate. So I'm interested in this phenomenon of differentiation of neural stem cells, their failure to differentiate, and how this can lead and arise to medulloblastomas. So I would like to focus then the rest of the talk specifically on group 3 and group 4 medulloblastomas and this question of identifying mechanisms of stalled differentiation as potential novel therapeutic targets. So here is a sort of a summary of our first pass analysis of the single-cell RNA sequencing data. And the first thing we did was to compare our medulloblastoma RNA sequencing to single-cell RNA sequencing of human fetal cerebellum produced from the Millen lab, recently published in 2021. We projected cell type labels from those developmental data onto our medulloblastoma neoplastic cells and identified both stem-like cells and more differentiated neuronal-like cells. So rhombic lip progenitors, The rhombic lip has two zones, the ventricular and subventricular zones. The ventricular zone is enriched for more primordial stem-like cells. The subventricular zone is enriched for proliferative intermediate progenitors. We identified cells akin to those in our medulloblastoma samples, but we also identified more differentiated neuronal cell types of the unipolar brush cell lineage. And this is a more differentiated phenotype which would arise from rhombic lip progenitors under normal developmental conditions. And so we want to try and understand what are the differences between the stem-like cells and the more differentiated cells. This is a differential expression test between the stem-like population and the more differentiated UBC-like population. What are the differences between these stalled cells, these cells that do not differentiate, and the more differentiated cells? Well, firstly, they're more frequently mitotic. And that's somewhat expected, although UBC cells do also divide in our data and in our analyses. But the other thing that came out was DNA damage repair. So these stem-like cells upregulate a robust program of DNA damage repair. And intriguingly, they also upregulate genes associated with telomere maintenance and telomere lengthening. This is critical because the mechanism of telomere maintenance in group 3 and group 4 medulloblastoma is unknown. All cancers require some mechanism to repair their telomeres. The telosmeros, the end cap of DNA, protects DNA. All cells, when they divide, the telomere gets a little bit shorter. And eventually, when it gets short enough, the cell should senesce and cease to further divide. However, all cancers have some mechanism to repair their telomeres so that they can keep dividing, but that mechanism is unknown for group 3 and group 4 medulloblastoma. I'd like to come back to that in a moment. So this is our attack data. This tells us what transcription factors are active in a particular condition that can be different from which transcription factors are differentially expressed. When we did that analysis comparing the differentiated cells to the progenitor populations, the most significant transcription factor which came up was this gene ZSCAN4. If you've ever done this type of motif analysis, this p-value is extremely small, indicating a very strong enrichment and a very specific activity of ZSCAN4 in these progenitor populations. It's interesting because ZSCAN4 is a gene from embryonal development. It's expressed in early embryos during zygotic genome activation, and it mediates telomere elongation in that context and DNA damage. It also protects the DNA from DNA damage during that process. So I'll come back to that in a moment, but I believe ZSCAN4 and telomere maintenance may be an intriguing therapeutic target for medulloblastomas. I'd like to tell you a little bit now about our genetic lineage tracing platform that we developed to try and test some of the hypotheses that we're generating when we look at the clinical specimens. We've developed technology to trace the lineages of individual cells using two approaches. Both are based on high complexity lentiviral libraries. And these libraries are high complexity. Each viral particle has a unique genetic barcode. That barcode is heritable. The daughter cells contain the same barcode as their parents. And it's expressed. It's an expressed polyadenylated transcript. So we can pick it up in our single-cell RNA sequencing reagents. We have two versions of it, one where the barcode is static, the other where the barcodes mutate over time because they're being continuously edited by a CRISPR process. We infect progenitor populations with this barcode library. Each cell gets a unique barcode that it can give to its daughter cells, and we can trace its lineage thereby. We pull out an aliquot for sequencing, inject an aliquot into the brains of mice, generate tumors, harvest the mature tumors, and then profile those cells. We generate 3 libraries from each experiment. One is the RNA library of endogenous gene expression.
Who?
The
Charlotte?
second.
A warning? Do we need to get?
What? Okay. Thank you. So we generate an endogenous gene expression library, but we also generate a library of cellular barcodes to tell how different cells are related to each other. We also generate a third library that's enriched for mitochondrial gene expression, because it turns out that mitochondrial mutations are a rich source for phylogenetic inference. This is an example of these data applied to a group 3 medulloblastoma model. In this diagram, each point corresponds to a cell. This summarizes endogenous gene expression. This second plot shows the clonality of each cell, the size of the lineage, the size of the family that each cell is drawn from. And the third plot gives an example of one particular clone, one set of cells that are related that have the same barcode. The interesting thing here is that all of the transcriptional phenotypes, stem-like nestin expressing more differentiated neuro-G expressing cells, all of these cellular phenotypes are achieved within one individual clone. So it's not different cells with different genetics producing different phenotypes. All phenotypes can be achieved within a particular given lineage. So we have methods for reconstructing phylogenies for individual clones. We look at expressed point mutations, in particular those in the mitochondrial genome. In our CRISPR system, the mutable barcodes also provide phylogenetic signal. And we can use that to construct a phylogeny of a phylogeny of genotypes. Now, you only ever observe the leaves of the phylogeny, so you don't actually know the phenotypes of the ancestral cells from which those leaves were derived. But we can infer those using a Bayesian method, and that's what we've done here. We've inferred the phenotypes of intermediate cells within the lineage of this one particular clone, and then we can now ask different questions about this. We can repeat this for all of the clones we observe, and then construct statistics on that summary, and that's what we've done here. And so this is sort of the baseline model. We now want to perturb it with ionizing radiation, different drug treatments. How do these hierarchies change and adapt once they're challenged with therapy? But even in this baseline model, we can answer basic questions such as, is there plasticity in medulloblastoma? Recent studies in glioblastoma have shown that there is a high level of plasticity. These tumors seem to have a rigid hierarchy akin to early development, but is there evidence of de-differentiation, transition from a more differentiated neuronal-like cell to a progenitor population? One of the things that I observed in these data, which I thought were very interesting, was first the composition. This first column tells you the composition of each clone. Each row corresponds to a clone. The yellow is the more primordial stem-like state. The green is the intermediate progenitor population. These are the UBC early cells, so early stage of differentiation, and these are the more mature cells here. Intriguingly, and the top box plot gives a summary of the column. Intriguingly, the composition of the model is very similar to the composition of the human disease, and that composition is roughly conserved at the level of individual clones. So the cellular composition of the tumor as a whole is recapitulated within individual clones. Now, when I first saw that, and we did that before we did the lineage, the phylogeny work, my first reaction is, well, there must not be any plasticity. Because how else could you maintain such homeostasis without a rigid hierarchy? However, that turned out not to be the case. Looking at individual clones, we can compare the phenotype of a particular clade to the phenotype of its subclade and ask, does this phenotype transition correspond to normal development, a maturation of the neuronal lineage, or is there de-differentiation taking place? And when we did that, we found that most cell types self-renew. This is the middle column, this sort of beige color here. This is the frequency of self-renewal. So most clades have the same phenotype as their parental clade. But some cell types transition, and the transition can go in either way. It can go to more differentiated or less differentiated, and the frequencies are roughly the same. So we do observe a high degree of plasticity, or a significant degree of plasticity. This is another way to summarize the data. We can look at the different cell types, the transitions between them, their frequencies, and statistics on those frequencies. And now I want to contrast this between control and treatment conditions. So in summary of this portion, human group 3 medulloblastomas and the model system that we used make the same cell types. And they make the same cell types in roughly the same proportions. And that those proportions are conserved down to the level of individual clones. We do find evidence for plasticity and dedifferentiation. And I think this poses a very important basic science and translational question, which is, how can these cells maintain such homeostasis in the presence of plasticity? I'd like now just to briefly touch on one other aspect that came up during this study, which is the focus on telomere biology, before I conclude my talk. And I won't have enough time to go through all of the evidence we've accumulated, but I want to just give you some of the basic ideas of what we found. So most cancers have mutations in a gene called TERT that drives telomerase reverse transcriptase expression and telomere maintenance. Most cancers have TERT promoter mutations, but group 3 and group 4 medulloblastomas do not. They're very rare. The other pathway of telomere maintenance is the alternative lengthening of telomeres pathway. The gold standard assay for assaying that is the C-circle assay. A recent study interrogated a relatively large number of medulloblastomas and did not find much evidence for ALT taking place. They did find ALT in a very small number of cases. This could be explained by an absence of ALT. These are false positives. But this could also be explained by ALT being a transient process that is only captured very rarely. We believe, and lastly, I want to say that this is a pan-cancer study of telomere lengths in different cancers. Medulloblastomas have some of the shortest telomeres pan-cancer, and thus they may be particularly sensitive to therapeutics that target telomere maintenance. In our data, we attribute telomere repair to a gene known as ZSCAN4. ZSCAN4 is expressed during early embryonic development, during zygotic genome activation. During that process, it decondenses telomeric heterochromatin, allowing for a transient alt-like process to take place. And for transient telomere repair, it positively regulates telomere elongation in developing embryos. It's added to Yamanaka factors when you want to make iPS cells because it enhances the efficiency and stability of the process. In our data, it co-expresses with stem cell markers like Nestin and CD15. It's upregulated in embryonal tumors compared to diffuse gliomas. When we knocked down ZSCAN4 using CRISPR inhibition, we saw significant amounts of telomere decay. When we overexpress ZSCAN4, we see significant telomere elongation. So ZSCAN4 positively regulates telomere maintenance in group 3 medulloblastoma as well as a group 3 model. When we knock it down, we find DNA damage occurring at the telomere specifically. The cells acquire a growth defect after around passage 6, consistent with telomere degradation. And when we inject them into the brains of mice, the tumors don't grow as fast. So I think ZSCAN4 and telomere maintenance is a good open problem. And I really like the way this study is coming together because it's an example of beginning with the genomics, the clinical specimens, progressing to the model system, and hopefully leading to a novel therapeutic target. Thank you for your time.
Thanks so much, Aaron. I think the way that Aaron concluded is perfect, right? We were hoping as part of this first session to really go all the way right from platforms and portals and programs to the level of detail, right? Analyzing single clones, essentially, at that level of detail in order to bridge across that. But that also lays a foundation for the challenge. But what Aaron just ended with has been core to the CBTN, which is that it often, almost always, begins with access to samples.